हेलो एवरीवन, वेलकम टू माय चैनल फार्मा एकेडमिया। इस लेक्चर से हम शुरू करते हैं स्टडी ऑफ मेडिसिनल केमिस्ट्री। जैसा कि आप सभी जानते हैं कि किसी भी सब्जेक्ट या टॉपिक की डीप स्टडी करने से पहले उसकी बेसिक क्लियर होना जरूरी है। इसलिए हम मेडिसिनल केमिस्ट्री की स्टडी भी बेसिक से ही शुरू करेंगे। जिसकी स्टडी के लिए पहले हमको ये जानना जरूरी है कि मेडिसिनल केमिस्ट्री क्या है, उसकी हिस्ट्री क्या है और ये डेवलप कैसे हुई। इसलिए इस लेक्चर में हम कवर करेंगे introduction, history and development of medicinal chemistry. So firstly, we will know the introduction of medicinal chemistry. The medicinal chemistry is the science which deals with the discovery and design of new and better therapeutic chemicals and development of these chemicals into new medicine and drugs. It means medicinal chemistry साइंस की वो ब्रांच है जो कि डील करती है डिस्कवरी एंड डिजाइन ऑफ न्यू केमिकल और थेरेपेटिक सब्सटेंस और एजेंट्स फॉर न्यू मेडिसिन और ड्रग डिजाइन. Okay, so now the question is arises, what is medicinal chemistry? Medicinal chemistry is one of the broad and important topic or subject in pharmaceutical science which involves the discovery of new chemical entities and systemic study of structure activity relationship of the compounds. Such study provide the basis for the development of better medicinal agents from the lead compounds found via random screening or systematic screening and rational design. So I hope you all may know all these terms: chemical entities, lead compound, structure activity relationship, and these screening techniques. If you know, that's so good. If you don't know, let me explain. So now, what is chemical entities? The chemical entities is a distinct substance or molecules with specific chemical or composition and properties. केमिकल एंटिटी वो सब्सटेंस या मॉलिक्यूल है विद द स्पेसिफिक केमिकल कंपोजिशन एंड प्रॉपर्टीज. ओके, now the structure activity relationship is the study of a chemical, how the chemical structure of compound relates to its biological or chemical activity, and जो help करता है साइंटिस्ट की understand करने में which part of a molecule are responsible to its effect, so they can design better drugs or molecule, a chemical, by modifying its structure. So essentially, SAR explore how small molecule or small changes in a molecule structure can lead to change in how it works. This is known as the SAR or structure activity relationship. Okay, the next term is the lead compound. Lead compound is the molecule that show the promising biological or chemical activity and serve as a starting point for further drug development and optimization. This is the lead compound. And these are the screening techniques. First one is the random screening. So what is the random screening? Random screening is the process which is used in drug discovery to test a large and diverse set of compounds for their potential activity against a specific target or disease. This is the overall process of random screening. And systematic screening. Systematic screening is the methodical approaches in drug discovery, where the compounds are tested in a organized or set step-by-step manner to evaluate their activity against a specific target or biological process. And the last one is the rational design. Rational design is the drug development strategy that uses the knowledge of the target's structure and function to create compound with specific desired effect. This is known as rational design. Okay. Now one of the scientists, Alfred Burger, he said that the great advance of medicinal chemistry have achieved by two types of investigators. So the one of the Those with the genius of prophetic logic who have opened a new field by interpreting correctly a few well-placed experiments, whether they pertain to the design or mechanism of action of drugs. And next one is the and those who give varied apparently the chemical structures of physiologically active compounds until a useful drug could be evolved as a tool in the medicine. The medicinal chemistry is the best of the definition is this interdisciplinary research area incorporating different branches of chemistry and biology in research for better and new drugs for the drug discovery. Okay, now come to the point origin of medicinal chemistry. Medicinal chemistry is the early investigation of natural products in the so-called pre-scientific era. So natural products having a history as a folk remedies were in use. So what is folk remedies? Folk remedies are the traditional medicine which are often natural treatment passed down through generation, typically based on cultural beliefs and practices rather than the scientific evidence. So for example, opium, belladonna, cinchona, bar, etc. So many drugs are originally used as a folk remedies. Nowadays have been abandoned. Abandoned means because of some resistance or may toxicity and all the properties because they may abandon it. So in the late 18th and 19th century, early 19th century, chemical experimentation led ultimately to its use in discovery of new drugs. In 1853, Henry, the scientist's name is Henry, so how convinced the idea that functional groups in natural product might be modified by chemical reagent. So he heated morphine with ethyl iodide, hoping to convert the alkaloid to codeine. So he obtained, however, new substance of quaternary salt of morphine. In the medicine in the antiquity, in means ancient era, Chinese medicine in 3500 BC. So Chinese herbs, some of the ingredients are still in use today. For example, reserpine, which is used for the blood pressure, high blood pressure, emotional and mental control. And another one is the ephedrine, which is used for the asthma treatment. And Egyptian medicine in 3000 BC, Papyrus Ibris 877 description and recipes given. Greek medicines from 700 BC. Illness is no punishment from the god. Medicine is considered a science. Diseases are due to natural causes and Hippocratic oath are given in this era. And the Roman medicine from approx 200 BC. Invention of hospitals, large influences of Greek medicine and materia medica, pharmaceutical description are given this era. Okay, so some of the years with their invention. So 1840s, ether, chloroform, and nitrous oxide move from the being part drug to anesthetics, being the research for other hypnotics. 1869, developed chloral hydrate, an oral compound that exhibit hypnotic properties, and one of the scientists F. Baer and co. market its first successful pharmaceutical sulfonol, hypnotic produces from the acetone. Based on sulfonol, von Mering suggests that a carbon with the two ethyl groups should be good hypnotic, make its diethyl acetylurea and then diethyl barbituric acid. These are the compounds used in the antipsychotic drugs class. Okay. So, in 1875, Carlebus isolated the salicylic acid from the Aspirea almeria and showed that it's an effective antipyretic. However, it is unpalatable and cause gastric distresses. Okay. So, 1883, von Nenke makes a salicylate ester with phenol that is called salol. Salol, it is a very poor solubility, but it is better tolerated. It is hydrolyzed slowly in small intestine to give salicylic acid, the first sustained release drug. Salol is the first sustained release drug. This is called phenyl salicylate. In 1890, Hoffman et al. Tested acyl salicylate acid and find it to better tolerated name it is aspirin. As in means aspirin in A means for the acetyl, A denoted for the acetyl, and aspirin for the aspiria. It is the rapidly hydrolyzed in a gut to give active salicylic acid. It is a prodrug. Planazone was synthesized in 1884, was the most popular drug worldwide until it was taken over by aspirin in the early 1900s. So, in the addition to being an antipyretic, it also cured headaches, and a new market was born. Okay. So, next topic is come the history of medicinal chemistry. Until 1900, digitalis, which is isolated from the plant digitalis, is used for the stimulation of heart muscles. And kinin alkaloid from the Peruvian bark, and it is used for the treatment of malaria and fever logging. And ipecacuanha, it is from the bark of ipecac and is used for the treatment of diarrhea. Aspirin from the meadow bark and against for the fever and pain treatment. Used for the pain treatment. Mercury is used for the syphilis. Okay. Since then, the early 1900s, synthetic drugs foundation of pharmaceutical industry, and since 1930, screening of natural products and isolation of their bioactive ingredients. Late 70s, development of recombination drugs, proteins and interferons, development of biotechnologies. In 2000, deciphering of the human genome, gene therapy, etc. Investigation of molecular basis of disease and future personalized medicine. And next, one of the biggest achievement for the field of medicine is the discovery of penicillin, which was found by the Alexander Fleming in 1928 at the fungus grave on bacteria plate containing staphylococci. So close to the fungus, all bacteria were killed. So biotechnology production of penicillin was stabilized during the Second World War. They can help serving the life of many soldiers. So this is the chronology of drug innovation. So we can see the complexity order. This was the accumulated knowledge of the human cell in molecular biology. Say the first one is the aspirin and sulfonamide, then antibiotics for antihypertensive, tranquilizers, anti-arthritis, beta blockers, then cancer therapies, etc. All the order of the drugs development, their invention. Okay. Then the drug discovery. Drug discovery is made up of two words, drug and discovery. So firstly we have to know the drugs. So drugs are the compound which is used for the prevention and treatment of disease. So what are the property for ideal drugs? So first one is the target biomolecule evolved in signaling or metabolic pathways that are specific to disease process by their protein or protein nucleic acid interaction and antagonist action inhibiting function of the disease causing proteins, inhibiting interaction of the proteins and activates other proteins that are deregulated in such disease like cancers. This is the modern drug discovery process. So they involve the six steps. So the target identification, target validation, lead identification, then lead optimization, preclinical phase, and drug discovery. Last one. This is this overall process containing at least 10 to 15 years for the drug discovery. Right. Okay, next one is the drug discovery technologies. Drug discovery technologies, first, these are the drug identification. It involves the genomics, gene expression profiling, and proteomics. And target validation, gene knockout or inhibition assay. Lead identification, high throughput screening, fragment based screening, and combinatorial libraries are involved in this phase. And Lead optimization, medicinal chemistry driven optimization, X-ray crystallography, QSR, quantitative structure activity relationship, ADME profiling, absorption, distribution, metabolism, excretion. And next one is the preclinical phase. It involves the pharmacodynamic, pharmacokinetic properties, ADME, and toxicity testing through the animals. And the last phase is the clinical phase, which involve the human trials. Okay, this is the overall drug discovery and development phases. So firstly, we have to know the identify disease and then isolate the protein involved in that particular disease. It will take a 2 to 5 year box. Then find a drug effective against the disease protein. Then preclinical testing and formulation of the drug and scale up the data and submit the data to file IND. And the IND will process for the human clinical trials. They can take the 2 to 10 years, and they collect the data and submit to IND. And then NDA. NDA. Then NDA will submit the data FDA approve for the FDA approval. So they will take 2 to 3 years. Okay. Then the future of medicinal chemistry. Future of medicinal chemistry. New drugs will discovered are invented by investigating human genomics and human disease genomics. Drug is the any substance presented for the treating or curing the preventing disease in human being or animals. So it may also be used for making a medical diagnosis or for the restoring, correcting or modifying physiological function. This is the oral introduction about the medicinal chemistry. If anyone have any doubts, so you can put in a comment box. Thank you.